but in most sloths. They are slow and solitary, and usually they are hanging in the trees sleeping. There are 2 species with 2 toes and 4 species with 3 toes. All species except the pygmy and the man sloth have been assessed to have low risk of extinction by the IUCN because of their wide distribution and presumed tolerance to habitat modification. Little is known about sloths' ecology and genetics, especially as they relate within species. Sloths are widespread in different habitats from Latin America, and subspecies or geographically isolated races may occur. Subspecies are usually ignored when designing conservation management plans, even though this diversity may reflect different responses to stressful situations like climate change. Accordingly, several questions have to be answered first in order to draft conservation plans. How came this genetic distribution about? Was it due to the environment or the climate? How many subspecies really occur within each sloth species? Could hybridization happen between species? How was, is, and will be the genetic diversity distribution in different climatic scenarios? My research focuses on answering these questions, but also takes ecological aspects into account. Subspecies can even further be distinguished by ecotypes, which are distinct populations adapted to the habitats and environmental conditions they live in. Ecotypes have to be defined based on ecological and genetic data. Environmental variables such as temperature or rainfall provide ecological data. Genes involved in local adaptation provide the genetic information. I will use museum specimens to obtain genetic data to determine the complete DNA sequence of 4 sloth species. But why museum samples? Because sloths have the exceptional ability to hide thanks to their algae-rich fur and slow movements. Besides, they are usually found in the upper forest canopy at heights 30 meters above ground, so they will be very difficult to find in the wild. This project will certainly provide the necessary information to fill knowledge gaps in sloth's life history, as it will be the first attempt to understand the historical mechanisms that have been responsible for their geographic distribution in the light of population genomics. Plus, it will certainly contribute to the definition of conservation areas and species conservation status.